so possibly muscle cells and bones, osteoblasts. So it all started something like 12 years ago, where during my postdoc in Berkeley, I I rediscovered, in fact, cells that were identified by Colmer and Agdur in these beautiful images from the book of Agdur in this case, where you have ciliated cells that are looking a little bit like hair cells from the ear, except that they are very disorganized, and they have one axon going on the basal side, while the apical extension here has, it seems, little microvilli that are called stereocilia in hair cells, and that are not really organized in a staircase, but more, a little bit disorganized in the cells. Colmer and Agdur had recognized that these cells were forming what they think must be a new sensory organ, but they had no way a century ago to really study those, with the tools we have now. And so they call this organ the Cine organ, as a parasagittal organ in the spinal cord, and they thought maybe the spinal cord has a third ear in the spinal cord. And where they disagreed very strongly was whether these neurons could interact with the Reissner fiber that I pointed in red here, a structure that is in the close vicinity of those cerebrospinal fluid contacting neurons, but not always in the fixed tissue. And so Reissner had identified the Reissner fiber in this little drawing from 1860, where he drew with a ruler a straight line. And this was a reflection of his observation in the lamprey, and it has been now found in rat, in the cow, in evidence again with electronic microscopy now in the lamprey, also in the trout or in the frog. And it's a big polymer whose diameter will depend depending on the species. So you can see in rat here, it's about 2 micron width, but it's much bigger in the cow. And the big question that we have is, what is the function of this Reissner fiber, and does it interact with the CSF contacting neurons in order to sense inside the spinal cord, and test possibly the idea of Colmer and Agdur that it could be sensing not really sound, but maybe mechanical deformation of the body axis. And so the Reissner fiber is made of a major protein called SCOspondin, whose structure you can see here. It's very conserved, and it's conserved across vertebrates, always with the same motif. It's almost 5 amino acids, so it's a gigantic protein. And it's acellular and outside the cells within the cerebrospinal fluid in the central canal. So when we started the lab, we were looking at the CSF contacting neurons and checked with Lydia Genoun, you can see here, if we could find these cells in different species. And we did find them in the macaque, in the mouse, and in zebrafish. And in the 3 species, we find that these cells are in fact inhibitory neurons. They express a synthesis enzyme and a transporter for GABA. And they also express a taste receptor that Charles Zucker had identified as a sour taste receptor involved in tasting changes of pH and osmolarity when expressed in the taste bud from the tongue. So it's a kind of a mystery already to understand, well, the cells are conserved, the 2 anatomists were right, but what could they be doing if they're expressing a taste receptor that is also found in the tongue? Could they be tasting the content of their cerebrospinal fluid? And if they do so, why would they taste the content of the fluid that passes the central nervous system? And the Reissner fiber, we can find it very effectively by using antibodies against Reissner fiber from cow. And so this is an immunohistochemistry from Yasmin Canto-Bellari, where you can see in zebrafish larvae the fiber very nicely labeled in the fixed tissue after using an antibody. So we started the team 11 years ago with another student, Kevin Fiedlin, who is on this slide. And we did together the very first experiments where we investigated the activity of these cells in vivo and saw what happened when we activate these cells using optogenetics. And we did that by using a specific line where we drive expression with the PE2L1 promoter. And by doing so, we have a very nice labeling of all these cells along the central canal that form like a chain, almost like a necklace, a straight line along the central canal. And if you look at them a little closer, you can find that the apical extension here is in contact with the cerebrospinal fluid in the central canal, and then the axon is running ascending and ipsilateral in the spinal cord, always going toward the head, so rostrally. And now in a section, you can find that these cells are very close to the motor neurons in green, and they are expressing different peptides. Here are the somatostatin positive CSF contacting neuron. And if we were labeling other peptides, like for instance from the family of the urotensin, we would label the ventral one who are not expressing somatostatin. So they're forming two rows, the dorsolateral left and right, and the ventral one that are labeled by urotensin-related peptides. So now if we look at the functional structure of these cells, what we see is that they are made of microvilli that form the apical extension with one cilium that has a 9+2 organization. You can see it in EM here. And this is typical of motile cilia. And so we can see that indeed in vivo, but also in vitro, the little cilium is beating. Even when there is no flow in vitro, the cilium is intrinsically beating. So we have a little steering mechanism in the apical extension, and then a very large surface area due to the microvilli that are actin based around the cilium. So with Urs Bohm, a German grad student who also started the team with me, we tested the hypothesis of Kolmer and Agder and figured out, well, are these cells indeed mechanosensory in vivo? And if so, how does it work? And so the way we designed the experiment is the following. We've been using an acoustic stimulus or just a puff pressure application of ACSF in the aortic vesicle. And when we do that on one side to an animal that has the head stuck in agar, we can elicit an escape response on the opposite side. That's what you see here. So a very strong tail bend on the opposite side. Because the animal is stuck with the head in agar, you get a single tail bend on the opposite side. And then we use a trick to understand how the cell responds to the bending on the right side here. We use a co-expression in these neurons of TAGR-FP to look at cell position and the GCAMP sensor. And by doing the ratio of the GCAMP to the TAGR-FP, we can correct for the motion artifact. Because the tail is moving and coming back into position, we have to adjust for typically 1 or 2 micron maximum. That can be due to the very large tail bend. And at the same time, we're tracking the tail so we can look at the fast response of the behavior and then quantify when the tail is bending on one side, do we see an activation of these cells and which of these neurons are responding? And so if we color code these neurons, the dorsal ipsilateral to the contraction, so on the concave side would be red, and then yellow for the contralateral side and purple for the ventral side. What we find, and it's all published in 2016, is that only the cells that are on the same side of the contraction, so on the concave side, are showing a calcium transient. And when we looked in mutant for which we knock out the PK2L1 receptor, the taste receptor to sour, we completely lose the response. So this was very weird because we were finding that the cell require a taste receptor to detect the change of curvature, and only on one side. So the way we took the next step was to think, well, okay, if they are mechanosensory and then they require the PK2L1 channel, let's look at them in using electrophysiology. So Jenna Stenberg, another grad student in the lab, came and put an electrode to record from these cells in vivo. And what she found, which is quite remarkable, to my knowledge, it's the only, single channel recording done in vivo, is that by just coming in these cells and recording in whole cell mode, she could find and observe those very beautiful, like single channel opening that you see here. And this is due to the fact that the cells are so resistive that they're typically 1 to 10 gigohm, that when you're recording in a whole cell, you have sufficient resistance to actually see the single channel opening with your whole cell patch clamp pipette. And that is associated with the fact that just an inward current of 10 or 20 picoamp is sufficient to elicit spiking in the cell. So a single channel is sufficient to elicit the spiking of those very resistive cells. And when Jenna recorded from the knockout PK12-1, she observed that suddenly there was absolutely no more channel opening in the animals in which we had removed this channel. Now, to demonstrate that they were mechanosensory, we had to look at them in vitro now and control the mechanical stimulus. And what she did with Patrick Delmas and Lucie Brosse, in particular in his lab in Marseille, is to push with a piezo against the membrane on these neurons that we have culture and done primary culture of. And she could find very nice evidence for the change of opening probability of the channel as soon as she was pushing against the membrane. So you do a 4-micron deflection on the membrane, and you see that the open probability of the channel increases. So instead of having 0 or 1 channel, she could have up to 3. And this is not the case in the knockout. In the knockout, we don't have the spontaneous activity. And when we come and push, we don't see any change of the open probability. So at this stage, we thought the following. We thought these cells must be a sensory cell that can detect the change of flow in the central canal. And we looked at the flow in very close detail, and without going into the model that we built and so on, what we discovered is that during a motor contraction, if the animal is not paralyzed in agar, the flow will be induced by the motor contraction. So here we have typical contraction going from head to tail, so rostral to caudal. And what we can see is that every time the contraction occurs, the particles that we have injected in the CSF are flushed from head to tail, from rostral to caudal, in the direction of the muscle contraction. So the central canal is acting a little bit like a peristatic pump, where when the muscle contracts around the body in the axial musculature, it flushes very fast the flow of cerebrospinal fluid. And exactly at the same time, when the muscle contracts and you see this motion artifact, what you see is a very clear activation of the CSF contacting neurons that are showing a large increase in GCAMP. So we thought we had understood, but we realized something was not matching. We realized that in this model, where the cells respond only to the change of flow, it should be symmetrical for left and right when the tail is bending on the left or on the right. And it should not be different for the two sides. We talked with François Galère and other people in EPFL who are experts of fluid dynamics, and they confirmed that if you look at the flow profile, it will be symmetrical left and right when the tail is bending, and 0 at the wall of the central canal, and maximum in the center, but symmetrical. So there was something missing. And the key came with the debate of Colmer and Agder about the Reissner fiber. So do they respond to flow? Maybe, but maybe not only to flow, because they can tell left and right. And so Pierre-Luc Bardet in the team came to generate the mutants of SCOspondin, and he used CRISPR-Cas to target really the first exon. And by doing that, he generated at first a null allele, ICM13, that you can see here. And he generated another allele by chance, ICM15, in which by chance there were 5 amino acids added to the protein. So no frame shift, only 5 amino acids added in the first exon. But it turned out that the first exon, this EMI domain here, had been shown in the 80s to be critical for polymerization of the SCOspondin into a fiber. And so Yasmine Canto-Bellarif investigated this in much more detail, and she used immunohistochemistry to look at the fiber and find that the fiber that you can see in wild type is gone in the null allele, ICM13, but it's also gone in the ICM15 when you have the 5 amino acid in the critical domain for polymerization. And what Yasmine Canto-Bellarif showed in addition is that in the three conditions, control, ICM13 and ICM15, if you look at the flow using beads, you find the exact same dynamic of the flow, dominated by Brownian motion at rest when the animal is paralyzed, with a bidirectional flow from rostral to caudal on the ventral side and opposite on the dorsal side, and with the same flushing that you see when the muscle contracts. So it's not that the flow is different, but now that we have tools with genetics to look at the role of the Reissner fiber, especially in the mutant ICM15, do we see a difference in the response of the cells to curvature? So now Adeline Horsdale-Imagine followed on that path, and she tested in control sibling versus homozygote mutant for ICM15 whether she had a difference in the response. And the difference was striking. It's quantified on the right. You can see that compared to the control sibling, the response to the muscle contraction is quasi-abolished in the mutant, where you don't have a fiber, but you still can detect SCOspondin material. So we have this model now that the CSF contact neurons are forming a little bit like Mickey Mouse ears, where they can detect left, right. And I didn't show you, but they can also detect ventral bending on the ventral side. And thanks to the presence of the Reissner fiber, they can asymmetrically tell if it's a bending on the left or on the right. And so we have two interpretations now that we're testing with Céline Bellagarda in the team. One is to see whether the fiber unable to enhance the flow gradient of CSF, because the flow will be zero on the surface of the fiber itself, or whether there is a direct contact between the apical extension of the neuron and the fiber itself. So this is really interesting because nobody knew what was the role of the Reissner fiber, and now we have a way to investigate the function of this sensory system, axial, made by the neuron and the fiber in vertebrates. And so we looked at it in more detail in zebrafish. And so this is the result of the tracking algorithm called ZebraZoom that Olivier Mira generated already almost 9 years ago now in the team. And so we looked at the effect of the abolition or the disruption of this sensory axis. And what we find is the following. So I will summarize again a result that we've published until recently with the work of Mingwei Wu. What we find is that in the mutant for PK2L1, and it's very similar but larger effect if we look at the effect of blocking the release from CSF contacting neurons with Botox, botulinum toxin, or if we ablate these neurons with two-photon ablation, we have a reduction of the tail beat frequency during those fast escape that the animal does to survive and avoid predator. And this is associated with a reduction of the power of the movement as well, where you see that in this case, it's ablation of the rostral-only CSF contacting neuron that leads to a reduction of the large tail bend called the C-bend that the animal does to swim away from the danger. And it's also slower, and it's associated with a reduction of the speed at which the animal swims. So this sensory feedback, and we've shown that it's the same for the classical DRG and trigeminal sensory feedback, unable to boost the amplitude of the movement, the speed at which the animal locomotes. And in the case of the CSF contacting neurons, it's doing that by direct projection onto the spinal cord circuit and the hindbrain. What is interesting is that it's not only affecting speed and power of locomotion, it's also affecting the posture of the animal, which is something we all thought posture is due to vestibular system. But when we silence, and in this case, this experiment is blocking the release of GABA and peptide from these cells with botulinum toxin, we find that the zebrafish larva, instead of swimming while staying horizontal, will start to roll over. And that's what you can see on this image. And in the work of Mingwei Wu, with the help of Olivier Mira, we developed an algorithm for analyzing with deep learning the posture of the animal and detect when it's starting to roll and the two eyes that are black here are overlapping. So we have a system, an axial sensory system, that can detect curvature in an asymmetrical manner and can control it actively, can control the posture during swimming. So we spend a lot of time, and I won't go into detail because it's not very fun, but we spend a lot of time to map the circuits because it's much easier in zebrafish larvae. The animal is all transparent. We can use optogenetics to activate the CSF contacting neuron. And then we can record with transgenic lines specific population of neurons, either the motor neurons in white here or the excitatory premotor interneuron or the command neuron, those reticulospinal neurons that come from the brainstem and project onto spinal cord. We also recorded from inhibitory cells and never found any projection. What we found is that the neurons in contact with the CSF are projecting directly onto motor neurons, as well as onto premotor excitatory neuron that control the slow swimming, and they make synapses on the axons of the command neuron that go from brainstem to spinal cord. So they have a way to gate information from the brain to the spinal cord to initiate movement. So this is like the work of many students that you've seen in the lab, together with Jeff Hubert, who was also a postdoc doing electrophysiology. And now the question that I'd like to take full advantage of in my time remaining is to see, well, could this system also be involved in other functions that enable the brain to communicate with organs in the body, in particular with musculature and the immune system. So it seems like a stretch, but I think it's the most interesting probably and relevant debate in this group is, the cerebrospinal fluid has been, shown to be very important for modulating neurogenesis and many aspects like migration of neuron, proliferation of progenitor during development. And it's also the site of pathogen invasion during an infection of the CNS. And so could it be that sensory neurons that are interoceptive, that are clearly inside the body, detecting parameters of the body, could they be involved in this process during development and also possibly detecting infection when the nervous system is under attack? So the first is the work of Yasmin Canto-Bellarif, who was a postdoc and now is a researcher who just got an important prize from CNRS last week. So Yasmin has developed the study of the developmental role of this axial sensory system and focused on the Reissner fiber. So she did the initial study of characterizing the mutant I told you about of SCOspondin, and what she observed that I didn't have a chance to tell you yet is that they don't develop a straight body axis. And so instead of being straight, these animals are in fact curled down, and they cannot form a nice straight body at the larval stage. They stay completely curled down like this. And so Yasmin has been investigating the mechanism and is now pursuing to understand how the sensory system is acting at different stages of development in the embryo, but also in the larva and in the juvenile. We had the observation with Jenna already that if you look at the knockout mutant for PA2L1, the sensory channel they express specifically, what you observe is that the adult has an hyperkyphosis with a rostral curvature on the ventral sides that you can observe here. And this is reminiscent of Sherman's disease in humans, which is much more occurring in men than in women. And it turned out to be the case in fish. We observe it mainly and more drastically in male adult fish compared to females. The second relevance of this sensory system in morphogenesis of the spine is apparent in the mutant for the receptor of the peptide that I mentioned in my introduction, the urotensin-related peptides are expressed only in the ventral CSF contacting neuron that are important for detecting the ventral bending when the animal is doing the equivalent of crunches. And remarkably, in the mutant for the receptor of those peptides, you can observe this really interesting phenotype that you can see here with like 3D torsion of the spine, which is reminiscent of idiopathic scoliosis. And what is really interesting is that in fish, like in humans, there is now a bias for female with the idiopathic scoliosis phenotype, 3D torsion of the spine. So this is the work of Yasmin Canto-Bellarif that she's now taking independently to understand the molecular and cellular mechanisms that are at play, and how by affecting this sensory system, you can effectively impact the body axis throughout life by acting on the musculature, but possibly on other targets in the body that she's looking into. Now, the second direction that I'd like to talk to you about is the direction of interaction between the nervous system and this sensory system with the immune system. When we had understood that the sensory system was important for posture, in 2014, I got a crazy idea that they could be maybe important for the stiffness of the neck that we can detect very clearly in the neck in adults. But if you look at people infected with that are diagnosed with meningitis in early stage, like infants or children, what you observe is that it's not only the neck that is stiff; it's actually the full body that become stiff, and the entire axial musculature is stiff. So we've been looking at this with Andy Prendergast and Hugo Marnas, who are represented here, and we looked whether this sensory system could be in fact important for detecting pathogen and possibly mediating this stiffness of the body during an infection. So we used Streptococcus pneumoniae, a bacteria that is the main cause of meningitis in humans. And we worked with a microbiology lab of Christina van den Broek in Amsterdam that generated different strains with different fluorescent protein and mutants for the bacteria. In this particular experiment, we used Streptococcus pneumoniae D39 with the M-cherry fusion to look at the propagation of the bacteria in the cerebrospinal fluid. And after 24 hours, we have a very nice colonization, invasion of the cerebrospinal fluid that we can see it very nicely here in the central canal. So we've wanted to have some hints of what these cells could be doing, and we started to look at the transcriptome by pulling many animals together. Even though there are only about 100 of these cells in each animal, we can pull animals together and sort them by fluorescence so that we cut the head off, look at the fluorescence of the CSF contacting neurons, and after sorting them, just look at their transcripts compared to the dark cells in the rest of the body. And what was remarkable is that in this transcriptome, we find that many receptors were present, not only the PKD2L1 that I mentioned, also the PKD1L2, that is an accessory protein that enables the receptor to be targeted at the membrane. We also observed multiple taste receptors that were orphaned from the T2R, the taste receptor that respond to bitter compound, and the T1R that respond to sweet, together with multiple peptides and receptors for the same peptide that are involved in innate immunity. And some of them are in fact markers of the CSF in the context of bacterial meningitis. Sorry, I just put my headphones because it starts to be really hard to have the echo. So we find that this peptide can be restricted to a single population, either the ventral or the dorsal population. And some of them were present in both ventral and dorsal. So we had some that were shown to be antimicrobial peptides, some that were shown to be markers of meningitis in humans, and some, like this one, in fact, is a protein that can be secreted in mouse and has shown to be attracting macrophage. So with this in mind, we tested whether we had a good model of meningitis in this zebrafish larva. And the way we did that is by injecting the bacteria in the cerebrospinal fluid in the ventricle, and we monitored first the behavior of the animal. And what we find is that most of the animals show what we call sickness behavior, when we don't locomote as much. And that's what you can see here in the pink. You have the animal are swimming much less than the control siblings. that are infected with PBS or injected with PBS. And what was remarkable is that in the few swim left by the infected animal, what we observed is that we had very different swimming, where we had very long swim events, and those were associated with a larger distance travel. And this is interesting because it occurred typically in 20% of the animal that we injected with the bacteria. And this typically in the zebrafish field is considered an epileptic-like seizure, where the animal, instead of doing a small, short forward bout, like you can see here, will have a very long swim that is very abnormal and not classical. Now, what was interesting as well is that in the animal where you inject Streptococcus pneumoniae, if you don't paralyze them with bungarotoxin like we did here to monitor the animal over time without having any motion artifact, if you let them, fully change posture, we observed something really interesting where the dorsal arching was elicited by the injection of the bacteria, and we didn't observe it in the control condition. And the curvature that we observe with this dorsal arching correlates with the amount of bacteria that we can estimate from the fluorescence that we have in the tail. So we have an interesting observation because this dorsal arching recalls a phenomenon of opistotonos that we can observe in the meningitis patient, where you start with stiffness of the neck, and if you don't get treatment, you will have the full dorsal arching with a very clear and abnormal posture. So the question was, are the CSF contacting neurons recruited during the infection? And could they be, participated to the immune response and the innate immune response, in particular of the host? And so what we observed with Andy is when we now have GCAMP again in these neurons, when we look at animal in which we have injected the Streptococcus pneumoniae in the ventricle, we have a very different activity pattern, where compared to the control condition where you see the flickering, a few cells like this one, and we'll have another one, I think, on the right in the video, a few cells are showing very large calcium transients that are much larger in amplitude, and they're also longer in duration than what we typically observed in the control. So if we quantify this data, it looks like this when you're looking at the delta F over F. We have flickering in the control, and then very large and long-lasting transient in a subset of the cell. And we did the experiment over 24 hours. We come back on the exact same cells every 2 hours, and we see that the same cells are showing the very large calcium transient, and with time we have gradually more cells showing those very large calcium transients. So it's really interesting. It shows that when we compile the data onto multiple infection, we have a reduction of the basal activity combined with an activation of a subset of the cell that shows this very large prolonged calcium transient that are both longer in duration and also larger in amplitude. And so we wonder, well, what could they do and how do they detect the presence of a bacteria in the CSF? And so for that, Hugo Marnaz did experiment in the lab where in vitro now, he's been testing, well, what happens if I puff different metabolites or cytolysine that are expressed by the bacteria and see if I can get calcium transient. And so he tested in particular pneumolysine here in orange and a mixture of bitter compound. And what was really neat is that compared to their spontaneous activity, or the fact that they don't respond in this condition to the pressure application of ACSF because it's far enough from the cell, it doesn't elicit a mechanosensory response, they could respond very nicely to pneumolysine with small calcium transient that we see at every pressure application. And they show even larger response to the bitter compound mixture, like you can see here, and repeated at every pressure application. So after Hugo had tested a mixture of the bitter metabolite that are just regular metabolites secreted by this bacteria in culture, he figured out which one were good at eliciting a response and found two compounds, in particular DMDS that you see on this slide, that can elicit very large response just alone when it's puffed on the cell like that. So we have indication that bitter compounds coming in fact from metabolites secreted by bacteria can act inside the central nervous system in a similar manner that what we can use them for in the tongue when you're tasting like, bitter milk after you have let the milk rotten on the table counter. You can see that the milk will change taste and it will tell you immediately that you shouldn't drink it. And we see that there is something very similar, in fact, inside the central nervous system in order to detect whether the CSF is corrupted. And then what happens when these cells are activated? So we've tested with different methods what was the role of these neurons in an infection. In these experiments, we use an enzyme called nitroreductase that we can use in combination with metronidazole to completely ablate the cells that are expressing it. And this is what you see on this image. With the BAS application of the metronidazole, we can completely remove the fluorescent from the nitroreductase and cherry. And so we have killed all the cells that were expressing nitroreductase. And when we do that, we observe that the ablation induces a reduction of the survival of the animals after infection, meaning that these neurons, the CSF contacting neurons, contribute to enhance the innate immunity of the animal. In order to dissect a little bit more the mechanism, we've expressed again the botulinum toxin light chain to block the release now without affecting the activity of the cell, without ablating the cell. We just block the release by acting on VAMP. And what we observed is that we have again an effect of the silencing of the release of these neurons that you can see on the survival here. When the CSF contacting neurons express botulinum toxin, we reduce the survival after 96 hours. And this is associated with an increase in the number of bacteria that you can detect in the body of the infected animal. So we investigate this a little bit further by doing transcriptome now in the context of the infection and find that upon infection, this neuron will express a new set of cytokine and complement factor that are induced specifically by the infection and can enable to boost even more the innate immune response. So altogether, I've been able to show you that the sensory system in contact with the cerebrospinal fluid has clearly different tasks. It's able to sense both the inner state of an animal and in particular whether there is an infection. It's clearly acting together with the Reissner fiber to detect the curvature of the spinal cord, and in particular whether it's concave or convex, sorry. And in response to that, it can modulate exploration and posture, but it can also impact the straightening of the body axis at early stage during embryogenesis and also later in life when the animal forms a full spine and needs to maintain the natural curvature of the spine. The last part I showed you is the work of Andy Prendergast, Hugo Marnas, and Kinky Jim. Is really this interesting interaction between the sensory system and the innate immune system in order to boost the innate immunity in response to infection. And it recalls the work of Isaac Chu on dorsal root ganglion neurons in the peripheral nervous system. So it seems to be a general principle that sensory cells are in fact involved in detecting pathogen in order to boost the innate immune response. And so I'd like to thank you for your attention, and I will put the name of the people, alumni, and current who are in the team who actively participated to this project. It was really an exciting adventure to test a new idea that we have a sensory system conserved across vertebrates in the spinal cord and find out how it's involved in multiple aspects of physiology and not only locomotion, but also posture and development and immunity. Thank you very much for your attention.
Thank you, Claire. Fantastic talk. Thank you so much. So we have one question now in our chat box, in our ask a question box. I will just read it to you.
Okay.
This is from Sylvia Henriques, and she says, very nice talk, Claire. Is the bending upon infection also unilateral? Does it mimic the PHD2L1 mutant phenotype? And if so, could there be a functional link between these two?
Yeah, so it's actually a very good question. Sorry, I have the echo again. It's a very good point. So in the infection, we see that the cells are recruited independently from their position, either the ventral or the dorsolateral left or right. And we're missing a piece here. We don't understand what makes a cell respond and not respond to the bacteria. So we're hoping to solve that puzzle using single-cell RNA-seq. There are just very few cells in each of those animals, so the experiment is really tricky to get to work. But it's a very, very important one. And then the second part of the question was going into the direction of, could there be a link between the mechanosensory and the chemosensory function of these cells? And indeed, this one is a very important one that I think we're still at the beginning of understanding. The lab of Brian Siruna in Canada has shown that you have indeed a link between inflammation and the deformation of the spine. And what we don't know, because it hasn't been studied in us in humans, to be honest, is whether in humans as well there is a possible link between being infected, during meningitis or encephalitis, and having deformation of the spine. We know in fish for sure that sick fish will tend to show deformation. So that's why we have to be very careful with the control being always siblings, because sick animal will tend to show curvature, deformation of the spine. And we know in human that it happens also in unhealthy, people. If you just do too much of the computer, you're going to be showing the dorsal arching like that. But it's not been studied whether an infection will tend to elicit deformation of the spine at the late stage, like those deformations that occur in juvenile or adults. And this is something very interesting that we're hoping to tackle in the team. I'm sorry, it was very hard with the echo to take any question during the talk at the end.
Sorry, do you hear me?
Yes.
Okay, good. So I was saying that I'm just going to ask you a follow-up question regarding the infection phenotype that you have, and I was wondering, when do you do the infection? So is this infection performed throughout life, or is it like an acute infection, right? So my question is going to whether this bending that you see is a developmental problem, or is it something that happens due to the infection, even if it's just an acute one? Do you see what I mean? So when do you do the infection? Is this throughout the life, or
no.
No.
We tested infection at the stage that was compromised. So we looked at basically the beginning of the larval stage. So we inject in 2 days old animal, and we monitor the infection throughout the days that follows. We had to find a compromise, and it would be very interesting to look at different stage now. The truth is that it's so tricky because you're dealing with different organisms, the bacteria itself, and the genetic background of the animal within a clutch that will be different. So you can't really test all the condition, and we had to find what is the response of the animal, of the host, when you're using different concentration of the bacteria. Focusing on that stage at 2 days old. And what is possible is that at that stage, innate immunity is indeed very important. And it could be that it changed over the life of the animal, where innate immunity becomes possibly less important. It's something that we didn't really look into. Our impression is that this sensory system is really tuned to be detecting bacteria and contribute to the, this dorsal arching. That's what we're testing now. But we would like really to look into other models to figure out if it's a general property. In particular, there are like efforts now to test for the role of these cells in mice. And so multiple teams, including I see Nicolas Van Averbeck, who is connected, and also Nicolo Zampieri. We also have Guillermo Lanoza in Argentina are investigating the connectivity and the function of these neurons in mice, and it's very exciting to see what they find.
Very, very exciting. Okay, so we have another question from Rita Figueiredo. She says, really interesting talk. Thank you so much. Do you think the prolonged calcium transients observed upon infection are required for the changes in movement? And do these changes in movement could mean an attempt to escape these bacteria somehow?
So our idea at this point is that the calcium transient that we observed in the context of an infection are completely different from what we observe in the spontaneous activity of the cell. And they are the best, way to get the neuron to release peptides. At this point, there is no evidence for difference in the snares between the dense granules that have the peptides and the clear vesicles. What is really different is the distance to the membrane. And so if you're looking at a clear vesicle with GABA in this case, they will be very close to the membrane, and they just need a small calcium transient to be released. In contrast, the dense granules, they are very far from the membrane, and they need much larger calcium to be fusing to the membrane. And so what we think is that those large calcium transients are ideal to secrete the peptides. And then the peptides that we find, they are involved in many physiological response of the neurons. And so they could be really involved for, maintaining the osmolarity of the CSF, changing a lot of the physiology of the animal throughout the body, and not just acting locally on the bacteria. But this is like, a complex question that we're very interested to tackle with people who are working in mammals as well.
Fantastic. So maybe I can ask you another question. So I was thinking about there's so much going on in the CSF and in the flux of the CSF, how these cells, the cilia move, how they connect to the Reissner fiber, right? And so I was wondering what is kind of the effect, or what could be, or if there is an effect actually in the motility of the cilia when there is an infection of the bacteria, or when actually the animal is moving. So is there alterations in the CSF constitution which then alter the actual the beating of the cilia because of sensory information that it's receiving, right? So
Yeah.
what comes first? Is it that the cilia control the flux of the CSF, or it's actually kind of the other way around, right?
So it's a very important point. No, the cilia of the ependymal cell in mammals and in zebrafish, they are called radial ependymal cells. They have glial and ependymal properties. So they are a little bit different, but in both cases, you have those cells that line up the ventricle that have very long cilium, and they are very motile. And they have, from what we quantified in fish, a very large spectrum of frequency at which they beat. And because they are very long, they're acting very effectively the same way that you need to beat when you do your mayonnaise. They are just very effective at steering. Okay. The cilia of the CSF contacting neurons are different. People have found that they are there also in mammals, by the way, on the apical extension, and we find them throughout the species we've looked at. they're short and they just act like this. So our idea, and we don't have, we haven't tested whether it's the case if you do experiments with particles in the flow, but they're actually not so good at generating the flow of CSF, but they're good at steering in front of the apical extension that acts a little bit like an antenna that can, smell or taste what is inside the CSF. And so you need to have a steering mechanism to be sure that you're moving particles around. So that's more our idea about what the cilia is involved in. And then the work of Niccolò Zampieri and his postdoc in Berlin has been showing very interesting data that if you remove just the cilium from these cells in mouse, you actually lose their modulation onto the spinal circuit, and you find similar defect than if you just ablate these cells. So it's a very interesting point that we couldn't test in fish.
Okay, fantastic. Thank you so much, Claire. We don't have any more questions in our chat box or in our ask a question box. So maybe we can just say goodbye to everybody. First of all, I would like to thank again Claire for joining us today. It was a fantastic talk. And I hope to see you all soon in our next seminar. So Claire, I would ask you to join us for just a couple more minutes, maybe 15 or 20 minutes.
Sure.
Into the Zoom chat, which I just shared, into the Zoom room, which I just shared in the chat. And we will be very happy to have people which wants to join us as well. So see you soon in a few minutes.
Thank you very much, Sita. Thank you, everyone.